Hello, everyone. Welcome to my Spring Science Symposium talk. I'm Zachary Gaoiran, and this school year I researched about the influence of water heater temperature on the quantity and pathogenicity of non-tuberculous microbacteria found in water heater sediment in Hawaii. So the genus Mycobacterium contains over 60 species and can be characterized by their very thick, waxy, lipid-rich hydrophobic cell walls that allow the bacteria to grow like fungus-like pellicles on liquid culture media. And mycobacteria can be divided into three groups based on a clinical criteria. The first group would be strict pathogens in humans and animals that are generally not found in the environment, which could include members of the Mycobacterium tuberculosis complex, or TB, and Mycobacterium leprae, which is the bacteria responsible for leprosy. And the second group would consist of mycobacteria potentially pathogenic in humans and animals that may become pathogenic in special circumstances. And some examples include NTM species Mycobacterium avium complex, or MAC, and Mycobacterium avium. And the third group consists of non-pathogenic species, which include other environmental NTM species. And so what are non-tuberculous mycobacteria, or NTM? Well, NTMs are mycobacteria found in the environment in everything from soil to dust to water. NTM are free-living bacteria and not obligate parasites of humans. NTM in soils and natural waters cause humans to be constantly surrounded by NTM. But although NTM are found everywhere in the environment, some species are not harmful to humans. And these species is called environmental NTM, which do not cause NTM-related infections. Unfortunately, only a small proportion of individuals are at an increased risk for NTM disease, but there are some species that are pathogenic and are harmful to humans. And these NTM, known as clinical species, cause NTM-related infections, including non-tuberculous mycobacteria pulmonary disease, or also known as NTM pulmonary disease. And NTM are opportunistic pathogens, meaning that they cause disease when a person's resistance to infections are weakened. NTM-related infections occur in immunodeficient and immunosuppressed patients, which are patients with a weakened immune system and have a reduced ability to fight off infections or other diseases. And this includes people who have had cancer or had a recent transplant, or any other illness that weakens their body's response to fight off infection. The risk of infection also increases for older people and people with a higher level of exposure to dust or smoke. Farmers, miners, and workers exposed to industrial dust are examples of workers who are at heightened risk of infection. In the United States, Mycobacterium avium, Mycobacterium intracellulare, and Mycobacterium avium complex are the most common NTM in infecting people. But the question is NTM pulmonary disease relevant to today's society, specifically in Hawaii? And the answer is absolutely yes. The incidence of NTM pulmonary disease is increasing in many parts of the world, including North America, while during that same time, the incidence of tuberculosis in industrialized countries had decreased. And what I found surprising was that Hawaii had the highest prevalence of any state in the nation, about 4 times the national average. And another study shows that Hawaii has the highest age-adjusted mortality rate from NTM pulmonary disease. On the screen, you can see the areas of Oahu, which is the island with the highest incidence of NTM that have the most cases per 100,000 people. In particular, NTM seem to be affecting populations different from others based on race or ethnicity. Asian Americans and Pacific Islanders were at increased risk of NTM pulmonary disease, independent of geographic area of residence, suggesting that race could be a factor increasing the risk of infection. A study showed that NTM isolation more than doubled from 2005 to 2013, with NTM isolation the highest on the island of Oahu. And the graph on the left shows that NTM isolation was highest among Japanese, Chinese, Korean, and Vietnamese patients. And the other graph shows that older people are also at an increased risk for NTM pulmonary disease. And so what does this data suggest? Well, first, it reveals the importance of genetics as a contribution to the increased risk for NTM infection. And further research is required to determine what those contributions are. But we can also learn how important the environmental contributions are for the increased risk of infection is. Soils in Hawaii are high in humic acid, which is a component associated with higher numbers of mycobacteria. And the increased environmental exposure, combined with a possibly more susceptible population, may be responsible for the higher prevalence of NTM pulmonary disease in Hawaii. Hawaii's unique environmental conditions may contribute to an increased level of environmental exposure through soil, but it may also increase exposure through water sources, and that is the focus in my project, which I focus on the topic of NTM in household plumbing. NTM can enter household plumbing, and the resistance to disinfectants and ability to attach to surfaces and form biofilms allow NTM to flourish in household plumbing. NTM are approximately 10 to 100 times more resistant to chlorine compared to E. coli. And because they're resistant to disinfectants, they have access to the available nutrients without any competition. And the biofilms they form are even more resistant to disinfectants. This prevents these slowly growing bacteria from being washed out of the system, and it allows NTM to flourish in household plumbing and persist in our pipes. And studies show that the DNA fingerprint of NTM recovered from NTM pulmonary disease patients were almost identical to those recovered from their showers, suggesting that NTM in their plumbing may be responsible for their infection. Water heating systems are especially an ideal habitat for NTMs with their warm waters and abundance of surfaces which allow biofilm formation. NTM numbers increase appreciably in water heating systems, which then allow higher numbers to enter household plumbing. But water heaters with temperatures greater than 130 degrees Fahrenheit were least likely to have NTM compared to water heaters with temperatures less than 125 degrees Fahrenheit. And researchers believe that water heater sediment that collect on the bottom have a resident population of NTM. And so with this, I wondered, are water heaters responsible for NTM in household plumbing? And if so, what part of the water heater has the most NTM? Although research suggests that hot water heaters have less NTM, there's a gap in knowledge and research about the content of water heater sediment. And with this, I came up with my research question, which is, does water heater temperature influence the quantity and pathogenicity of NTM found in water heater sediment in Hawaii? In order to answer this question, I partnered with local professional plumbers to collect sediment and swab samples from used household water heaters. The pictures on the right show the sediment found in water heaters, and in total, I was only able to collect 6 samples. Due to the pandemic, it was difficult to collect samples from fresh water heaters that haven't been sitting for a while, which limited the amount of samples that I collected. And the temperature of the water heater was collected, and a survey was conducted to determine some information about the heater, such as the water heater's age and if the water heater has been previously flushed. Then I plated the samples and incubated it for 21 days, and I took pictures on day 1, 4, 7, 14, and 21 to monitor the bacteria growth on each plate. And so here are some of the examples of the plates I cultured. The top row of pictures is a neat sample, which is the untreated sample that we plate all the bacteria that we collected from the swab or in the sediment. And so that's why you see so much bacteria. And the bottom row of pictures is a CPC treated sample, which kills all other bacteria except NTM, leaving only NTM to appear on the plates. And although I only collected only 6 samples, there was quite a bit of work that had to be done. And I plated about 60 plates, and I took about 300 pictures of the plates. And so after 21 days, the next step was to identify the bacteria that grew on the plates to see if there was NTM in, or if there wasn't, and if there was NTM, if the NTM was pathogenic or not. So the first thing we had to do was extract the DNA as shown on the screen. The bacteria was first scraped off the plates and transferred to nucleospin B tubes, where we prepared the samples. The samples were then lysed by incubating it at 70 degrees Celsius and vortex. Then we had to clean the DNA by removing any contaminants and filtering the lysate. Then we had to bind the DNA and wash and dry the silica membrane. And after this step, we had to elute the DNA to complete the DNA extraction. Next, we had to use the 16S gene. We sequenced the 16S gene. First, we did a PCR to amplify the 16S gene using a thermocycler. Then we had to concentrate the DNA by using a magnet to pellet the DNA and wash it off with ethanol. Then we used a qubit fluorometer to quantify the DNA. And finally, we loaded the DNA into the MinION fungal flow cell and analyzed the data using MinNode data analysis. And so here are the results. In total, like I said previously, I only collected 6 samples, but only 4 of the samples I was able to sequence because the DNA from the other 2 did not amplify well. As you can see in the diagram on the left of the number of cumulative reads for each taxon sequence, there were quite a bit of different bacteria in the sediment samples I collected, shown by the large number of different reads. And so this shows that there is a diverse population of bacteria that live in the water heater sediment in my samples. However, you can see that there isn't, or there's zero reads for mycobacteria in the samples I collected, suggesting that there were no NTM in the sediment samples I collected. And shown on the right is a taxonomy tree showing the different genera for all four samples and the relationships between each other. Although I do not have enough samples to confirm my findings, we can see that the early results show that there are no NTM in water heater sediment in the samples I collected. And the early results do not support my hypothesis because I do not find any NTM in the sediment, regardless of the water temperature. And I'm unable to conclude if water heater temperature affects the quantity and pathogenicity of NTM found in water heater sediment. I found this data surprising because I expected there to be a large number of NTM, but for further research, I will collect more sediment samples from water heaters, and I may also look at other components of household plumbing that may have NTM, such as shower heads and faucets. Another idea that a judge told me about at the district science fair was to introduce our own NTM in a sample and run that sample through my protocols to see if we end up with NTM at the end when we sequence it and to see if my protocols work. But throughout this experience, I definitely learned a lot of valuable skills. I learned basic lab techniques and skills that will be very useful for me in the future. I learned the importance of organization, especially in a large project like this one. And I learned that research is a process that takes a lot of time and there's a lot of obstacles that you will face. My advice for future research students is to ask questions, read as much as you can about your topic, and most importantly, stay organized throughout the entire process. Finally, I would like to thank my mentor, Dr. Jennifer Honda, for her guidance and support throughout the entire process. I would also like to thank Matt Yoshimura and Ronan Numasaki for their help in collecting samples, and Dr. Chan for overall helping me with the research project and helping me in the lab. Also, I would like to thank Dr. Cottrell, Mr. Key, and Mr. Gushikin for helping me to get my project started earlier in the summer with my initial plan to be able to collect samples. And finally, I would like to thank Papa Jack for his support with the K Fellowship, which allowed me to do research over the past summer. And so here are my references. And yeah, that's all I have today. Thank you for listening.